I'll rush through the topics today about antigen antibody. So about antigen I'll be taking right now. So it has got you know what's an antigen is, right? We have studied it during our histology and everything, the parts of the antigen and everything. So I'll just rush through the topic right now. So what is antigen? This has got two main properties. That is, one is immunogenicity and one is immunological reactivity. What do you mean by immunogenicity is the ability of an antigen to induce an immune response in the body. That is, it stimulates the bone marrow or the thymus or the primary immune response to produce antibodies. That is antibody production. That is what immunogenicity means. Whereas what is immunological reactivity means, it's a reaction. That is antigenicity. It's the ability of the antigen to react with the antibody, and they form a antigen-antibody complex. And the test which we use for finding the immunological reactivity or the antigenicity is precipitation test or an agglutination test. So what is a hapten? So hapten is same like antigen, but it's actually a defective antigen. Why it is defective? Because it can retain antigenicity or immunological reactivity, but it does not induce an immune response. Only thing is that it can bind to an antibody, but if you give a hapten alone inside the body, it won't induce an immune response, or it will stand there only. But once the induced response is stimulated, then it will bind to an antibody. That's why it's called as hapten. It's a type like an antigen. It has got one single property of an hapten, that is antigenicity or immunological reactivity, but it does not have immunogenicity. So it's a low molecular weight molecule that lacks immunogenicity. That is, it can't induce the immune response, but it retains antigenicity or immunological reactivity. That is, it can bind to a specific antibody. But hapten can be made immunological reactive by it can induce an immune response by binding to a large protein molecule called a carrier protein.
Yes.
So what is the classification of hapten? So these haptens are classified into two types, mainly simple hapten and complex hapten. What do you mean by simple hapten? Means it has only one epitope. You know what is an epitope?
Not exactly. So could you just
epitope is a point, is a receptor on an antigen where an antibody binds. So on an antibody, there will be a specific receptor called paratope. So this epitope and paratope of the respective antigen and antibody is the one which binds. Got it? So epitope is present on an antigen, whereas paratope is present on the corresponding same site is paratope on an antibody.
Antibody.
Okay? Yeah. So only in simple haptens there is only one epitope, and it doesn't precipitate, hence it can't be visualized. Whereas complex haptens are made up of two or more epitope, and since it's made up of two or more epitopes, it's a large molecule, it precipitates, hence it can be visualized. So again, I've written a short note about epitope. Epitope are the smallest antigen determinant that binds to a specific part of an antibody, and that specific part of an antibody is the paratope. What are the factors influencing antigenicity? This is actually a 5-mark question. So what are the factors which influence the strength of an antigen or the immune reaction caused by an antigen? That's what's called antigenicity. So antigens is the one which stimulates the immune response.
Immune response.
Yeah. What are the factors which influence the antigenicity? So what size of the antigen, then chemical nature. There are different factors. So I'll explain to each and everything about it. Size of the antigen, how does it influence? Larger the size, more is the antigenicity, that is more potent. Then next is chemical nature. In chemical nature, that is the thing. Proteins are more antigenic than carbohydrates. Then carbohydrates are more antigenic than lipids, and lipids are more antigenic than nucleic acid. Remember this order, okay? They might ask you in Viva, what is the chemical component which is more antigenic? That is, proteins are more antigenic than carbohydrate more than lipids, and lipids more than nucleic acid. Then susceptibility to tissue enzymes. Antigen is degraded by tissue enzymes, leads to generation of more number of epitopes. So once the antigen is degraded, it leads to more number of epitopes, and this will increase the antigenicity. Whereas, so example is lactase is not degraded, so it does not induce antigenicity. Whereas L amino acids, there's a proteins there, are antigenic, hence degraded by tissue enzymes. The next, which factor influences the more is the foreignness. So how foreign the antigen is, or the one protein is, if it's very foreign, then it will induce more severe antigenicity. More the foreignness, more the antigenicity. So you got till now, right? Understood till now?
Yes, sir.
Okay.
Yes, sir.
Next, other factors are genetic factors. Uh, Genetic factors is different individuals of a given species, so different type of immune response. This is very classical. Like now, I'll give you a classic example: COVID. See, one person response to the COVID is different for different person. So I might
Yes.
see, you might since you have bronchial asthma, you might have a severe COVID. You might, I'm telling you. But since I don't have bronchial asthma, I might not get COVID as severe as yours. So the response of our body is different for different, so for the same antigen itself. So that's how genetic factor works. Different individuals of a given species show different type of immune response towards the same antigen due to genetic differences between the individuals. The next is optimal dose of antigen. Now dose also different. Before I told you about foreignness, just now it's about dose. So if the small dose of antigen enters into the body, it might not elicit like small quantity of viral common cold enters. You might just neglect. You might have a slight cold and just go off. But if very strong quantity of like common cold virus size or rhinovirus enters in your body, you might get a severe cold. That's what it is.
Yes.
Antigen is immunologically active in optimal dose range. There should be a particular dose range for to elicit a immune response. Too little dose fails to elicit immune response. Too large dose leads to development of immunological tolerance. That's it. Sometimes, so very large doses will cause very severe immunological reaction. So what then next is route of antigen administration also. The immune response is better induced by parenteral administration. That's IV route causes more severe route of what antigenic stimulation. Same, you just you take a Rantac tablet or some paracetamol tablet, you might not get allergic reaction. But the same content, if you give through IV route, the person might get an anaphylactic reaction. Or even if you take a diclofenac, diclofenac is the best example because when you start practicing in internship, you'll find out a lot of diclofenac reaction, allergic reaction. That's why the nurses they give the IM. IM if you give, sometimes they might get allergic reaction. Sometimes they might give IV also in 100 ml. Got it. So that this is the route of antigen administration is very important in causing an allergic reaction. The next is repeated dose of antigen. To generate an adequate immune response, repeated dose of antigen over a period of time is required. So some patients, they might a small dose of, like if you might have some people have food allergies, especially to prawns. So he might have a smaller quantity of prawns. It might not elicit a allergic reaction. But the same person, when he has like one plate of prawns, before he had only one spoon, but now he has a one plate of prawns. That time it elicits the allergic reaction. Got it?
Yes, sir.
So repeated dose of antigen will also is a factor for causing antigenicity. So these are the main 7 factors: that is size of the antigen, chemical nature, susceptibility of tissue enzyme, that is breakdown of the antigen causes increase in number of epitopes, which causes increased antigenicity. Then foreigners, then genetic factor, optimal dose of antigen, then route of antigen administration, that is the IV might cause more severe than the per oral or inhalation route. Then repeated dose of antigen. The next is the antigen host relationship. That is antigen and the body's relationship. Host where the antigen enters the relationship. That is self or auto antigen. That is sometimes the self, that is our own body may act foreign to our own body, and so like as the autoimmunity which happens. Autoimmune reactions. That is the self or auto antigen. It belongs to the host itself. It is not actually immunogenic, but it shows immunological tolerance suddenly, as the immune system doesn't react to its own antigen. So this self or auto antigen actually it is the antigen which is present in our body sometimes may not react with our own body's immune system. That is why it's self or auto antigen, and it shows immunological tolerance. The next is non-self or foreignness antigen, that is immunogenic antigen. That is, there are 3 types. There is alloantigen, that is, it depends on the species. That is species specific. Then isoantigens are present only in subset of species. Example, the blood group antigen and histocompatibility antigen. That is, histocompatibility antigen is actually used in transplantation. That's why they do HLA typing before transplantation, that's between the receiver and the donor. They do a screening. That is like HLA antigen, histocompatible antigen. Then next is blood group. Blood group antigen, you know, right? O positive can be given to O positive, AB positive can be given to AB positive lab, AB negative lab. That's also is a iso antigen. Then next is heteroantigens, antigen belonging to two different species. That is antigen of plant or animals or microorganisms. These heteroantigens, no, you might find like how it is important because sometimes we use a certain test. Like I'll come into it in the further, where we use a test of what antigen or antigen of a species to find out the antigen of a different species. So heteroantigen is important in that way. Then it is what is heterophile antigen? Is a type of heteroantigen actually. That's what heterophile antigen. It's a type. It's a subdivision of heteroantigen. Cross is a cross-reactive antigen. If present in two different species, this is what the importance of heteroantigen. That's what I told you right now. So it is a cross-reactive antigen. That is cross-reactive means antigen of one species will bind to the is used for the detection of the antigen of the different species. That's why it's called cross-reactive antigen. It's present in two different species, but they share the same epitope. Epitope, I told you, is the receptor over the antigen, the antibody binding site. And these two antigens will have the same epitope. So that's why they help in identifying the different antibodies. So antibody produced against one antigen of one species can react with the antibody of the different species having the same type of epitope. Example is this is okay, the Wales-Felix test, the Paul Bennell test. So in Wales-Felix test, what we do is the antibodies against typhus fever. We use the rickettsial antigen using protease antigen. What is typhus fever is caused by rickettsia. So the antigen against rickettsial antigen, antibodies against rickettsial antigen is used by detecting using a protease antigen. So here the antigens are rickettsial antigen and the protease antigen. Rickettsial antigen will elicit an antibody reaction, and this antibody will bind to protease antigen. So if it binds to protease antigen, the Wales-Felix test will be positive. So that's why how we find out that the patient has typhus fever.
Yes.
Got it? Understood?
Yes.
Or you are confused again?
So just the example, can you just
okay. See, in Wales-Felix test is a test for typhus fever. Typhus fever is caused by an antigen called rickettsial antigen. So we have to diagnose that rickettsial antigen, but we can't find we don't have any test to diagnose the rickettsial antigen. Only this test is available. So how do we do this test? We put a protease antigen in the serum of the patient. Okay, you'll find an antigen-antibody reaction. It is that test will be you'll find clots formation in the test.
Yes.
So the test is positive for Wales-Felix test.
Yes.
Yeah, but did we do the test for protease disease? No, right? We did a test for typhus fever.
Typhus. Yes.
So what typhus fever has what type of antigen? Rickettsial antigen, because it's caused by rickettsia. So it's a rickettsial antigen. But we didn't put rickettsial antigen and find out the reaction, clot formation.
Yes.
But we use protease antigen.
Protease antigen.
So here the rickettsial antigen epitope and protease antigen epitope is same.
Same.
That's why we use protease antigen to find out the antibody against typhus fever.
Yes.
So here the heterophile antigens are protease antigen and rickettsial antigen.
Okay.
Now you understood?
Yes, sir. Yes.
Okay, fine. Same in Pond Bennett test also is same like this, something like this only. Infectious mononucleosis is caused by Epstein-Barr virus. Here the antibody is detected by what? We don't use Epstein-Barr virus antigen. We use what? Sheep RBC antigen. So if the sheep RBC antigen binds to antibodies against Epstein-Barr virus formed against Epstein-Barr virus, you find clot formation.
Clot formation.
So here the sheep RBC antigen epitope and the Epstein-Barr virus antigen epitope is same. That is the heterophile antigen.
Yes.
Got it?
Yes.
Okay. Same cold agglutination test also and Streptococcus mycoplasma MG test also. Same principle.
Yes.
So this streptococcal or mycoplasmal MG test is used for primary atypical pneumonia. Then for CAT test, human O blood group RBCs are used. For MG test Streptococcus, MG antigens are used. MG is miliary group antigen. There's a few examples. You don't need to know every one of them. You just need to know veil flexus, Paul Bennett's, chlorolactic test. The same, the principle of this test all, no, when it comes to individual topics, I will again explain to you how this test is done.
Yes, sir.
Okay, fine. You just need to know the names right now and what are the antibodies you formed against which type of disease and what antigen we use. As just for example,
Yes.
for heterophile antigen, no, they might ask you for like 3 marks or 5 mark questions. You have 5 mark questions, right?
Yes, sir.
Yeah. So you have 2 marks, 5 mark, and 10 marks, right?
No, sir. We have 15 mark, 8 marks, and then 4 marks and 1 markers.
Yeah. For 4 marks, no, they might ask you this question. It's because it's a short nose heterophile. Everybody knows that heterophile antigen. They'll forget. They'll get confused.
Yes, sir.
It's an important topic for you in 4 mark question. Okay. So now we'll come to adjuvants. You know what are adjuvants? Adjuvants are any substance that enhance the immunogenicity of an antigen. You know what is immunogenicity?
Yeah, immunogenicity is the ability to produce an immune response.
Yeah, it elicits the immune response. And antigenicity is actually antigen and antibody binding.
Binding.
Yeah. So what is adjuvant? Adjuvant is a substance which enhances the immunogenicity of an antigen, usually added to vaccines to increase the immunogenicity of vaccines. So that's adjuvant is just an additive actually, and it increases the immunogenicity of an antigen. So what is the example? What are the examples are? You know, right? DPT vaccine. Then
Yes.
For the influenza vaccine. Then next is like for DPT vaccine, the adjuvant is actually pertussis. Pertussis acts as an adjuvant for the diphtheria toxin and the tetanus toxin. And the DT and the diphtheria toxin and tetanus toxin also may act as an adjuvant for the influenza virus vaccine. Then some viruses may have alum and mineral oil. Then next is this one called Froun's adjuvant. It's a complete and incomplete adjuvant. This you just need to know the name. Then Mycobacter bovis also might be used as an adjuvant. Then Silica beryllium and thiamercel also might be used as the adjuvant.
Yes.
Then next is biological classes of antigen. Depending on the mechanism of inducing antibody, antigen are classified into T cell dependent antigen, T cell independent antigen, and super antigen. I'll come into the next slides where they'll explain about why it is called T cell dependent, why it's called T cell independent, and super antigen. That's why they told the principle is that depending on the mechanism of inducing the antibody, the way the antigen induces the antibody, it is classified into these 3 types. So now I'll tell you why it is called T cell dependent. T cell dependent because the antigen is taken up by the antigen presenting cells. APC means antigen presenting cells. So friend, I'll be using APC, then it means that it's called antigen presenting cells. Even in books also you might find it APC way. So what is APC? Is antigen presenting cells. Then this antigen presenting cells, why they call antigen presenting cells? Because they present this antigen to the T cell.
T cells.
So once they present to the T cells, this T cell is gets activated. Once T cells get activated, they secrete cytokines. Cytokines like interleukins 6, interleukins 1, tumor necrosis factor, tumor necrosis factor alpha, tumor necrosis factor beta. All these are interferons gamma. All these are cytokines. So once these cytokines are released, they stimulate the B cells. And once B cells are stimulated, they get activated. They convert into plasma cells, and then they produce these plasma cells are the ones produce the antibodies. Got it?
Yes.
Yeah, and they are called T cell dependent because it is depending on these cells are
T cell activation.
pathway has a T cell activation.
Yes.
Okay. Now next is T cell independent antigen. Here the antigen direct surpasses the antigen presenting cells and T cells, and they directly bind to the B cells, and they cause the activation of stimulates it polyclonally. That is, it converts into plasma cells by after antigen binds to B cells, and then they cause the immune reaction. It's not, and here it is non-specific binding. Hence, there is increased secretion of non-specific antibodies. Here, the binding is actually non-specific. It's not like a specifically to a particular antigen. So it is here the antigen binding is non-specific, and that's why the antibodies are not specific to a particular organism. So it's non-specific antibody. That is, there is hyper gamma globulinemia protection.
Yes.
So next is super antigen. Why they are called super antigen? Because here they directly binds to the variable beta region of T cell receptor. Now the variable region, what is the variable region? I'll tell you. In antigens, there are 2 regions. There's variable region and constant region. So variable region is the area where which will have the paratope. the antigen binding, antibody binding site. So again, I'll be telling you about the structure of an antigen and the antibody. So that time I'll tell you where it is. Again, I'll explain to you each and everything. So it directly, so remember that super antigen, no, there is no antigen presenting cells. Directly it is binds to the variable beta region of the T cell receptors. Okay, and they are not processed by the antigen presenting cells. And then since antigen presenting cells they are not what processed, hence again the binding is non-specific. And here no, once it binds super antigens, once it binds to the T cells, they cause massive, very massive cytokine release, and there is no control over it because it is non-specific binding. So they cause massive release so that it kills all the bacteria or viruses or the foreign bodies in our body. But there is a negative effect because of that. That is, it causes massive release of cytokines, causing cytokine storm. And because of this cytokine storm, no, it causes diseases called like Staphylococcus scale skin syndrome, the toxic shock syndrome. You read Staphylococcus chapter?
Yes, for example, I don't remember exactly.
Okay,
but,
yeah, that's a toxic shock syndrome. The patient goes into shock because of this ultimate high cytokine release. That's why sometimes the antibodies are good, sometimes the antibodies are very bad. So I told you what are the inflammatory mediators. Inflammatory mediators like interferon gamma, interleukins 1, interleukin 6, tumor necrosis factor alpha, and tumor necrosis factor beta. So now next is polyclonic activation of B cells, and the B cells converted into plasma cells, and plasma cells causes the production of non-specific antibodies, and there's hypergamma globulin. So this is the flow chart. This is the first one you see. The antigen is present to the antigen presenting cell. This is the T cell dependent first one is. But then antigen presenting cells presents the antigen to the T cells, and T cells presents the causes release of cytokines. Since it is a specific binding, no, it releases only specific needed cytokines, and its cytokines causes stimulation of B cells, and B cells gets converted to plasma cells, and plasma cells causes antibodies. Second one is super antigen. That is, there is no antigen presenting cells, or it is surpassing. They wrote that bypass antigen presenting cell processing and binds to non-specifically to T cells. And T cells causes release of massive non-specific cytokine release, which causes chronic activation of B cells and then hypergamma globulinemia. Then last one is the T cell independent antigens, that is directly present to the B cells and non-specific binding, and B cells get activated into plasma cells and they causes hypergamma globulinemia. So this is a flowchart. You can just go through it before the exam. You will remember this flowchart.
Yes.
Then next is what is the examples of super antigens? Super antigens, especially bacterial super antigens like Staphylococcus toxins, like toxic shock syndrome toxins. That's what I told you, Staphylococcus. Exfoliated toxin enterotoxins. Then it's Streptococcus pyrogenic exotoxins. Then Yersinia enterocolitis, Yersinia pseudotuberculosis. Then viral super antigens like Epstein-Barr virus causes associated super antigens, cytomegalovirus virus super antigen. Then fungal super antigens is Malaysia purpur. Just need to know the names, no problem. Just need to know the what examples. Super antigen again is a favorite topic of microbiology people. They'll ask you for 4-mark question, or they might ask you for a short note also. So what are diseases associated because of this? What antibodies produce? Super antigens. What are diseases associated with this? Toxic shock syndrome toxin produces toxic shock syndrome. Then enterotoxins causes food poisoning. Then streptococcal pyrogenic exotoxin A and C causes scale skin syndrome also. Then the other examples atopic dermatitis, Kawasaki syndrome, psoriasis, etc. First 3 examples you must know: toxic shock syndrome, syndrome, food poisoning, and scale skin syndrome. These are 3 important topics of the where you find it because super antigens you get the massive cytokine storm. In Staphylococcus also chapter they might ask you a short notes on toxin. I think this time they ask you, I guess, what are toxins of Staphylococcus.
Yes, sir.
Right? Yeah, that's a favorite topic. Anyway, during the skin topic when I teach you, I'll again be telling you individually about different toxins.
Yes, sir.
Then next is I'm telling you about antibody right now. So antibody or immunoglobulin is a specialized glycoprotein. It is produced by activated B cells. Then activated B cells are called plasma cells. That's what I told you. Once the B cells get polyclonal activated, they convert into plasma cells, and it's the plasma cells who produce the antibodies. So produced by activated B cells called plasma cells in response to an antigen is capable of combining with the antigen that triggers its production. So there are 5 classes. There are 5 classes of antibodies. They are also called immunoglobulins. immunoglobulins G, immunoglobulins A, M, D, and E. So this is a generalized structure of an antibody in front of you. So that's what I told you. There is a light chain and a heavy chain. Light chain is on the outer side and heavy chain is on the inside side. But sometimes some diagrams might show light chain in the inner side also, doesn't matter. But these chains, no, they have on the antigen binding side, that is the variable region. You saw the purple color, that's the variable region, and the rest of the regions, which is light blue, are the constant region. Constant. And variable region will have the amino end, and constant region will have the carboxyl end.
Carboxyl.
Yeah. And these two light chains and heavy chains are bounded together with a disulfide bond.
Yes.
So that per brown color two bonds as a disulfide bond. And you know on the next side there will be a region called the hinge region, which has the antibody like a you know what is between the door and the wall. There's a hinge, right? It helps in the movement. So now a structure of antibody is a Y shape, is a heterodimer. It is composed of 4 polypeptide chain. There is a 2 light chain and 2 heavy chain. It is a light chain molecular weight of 25,000 Dalton each, whereas heavy chain is about 50,000 Dalton each. So 2 light chains are actually there are 2 types of light chains. There's kappa or lambda. It might be either both the light chains will be kappa or either both the light chains will be lambda, but never it will be like kappa lambda together. Yes. Either it will be kappa or it will be lambda. The next is heavy chain. There are 5 class based. I told right 5 classes of antibodies are there. 5 classification of antibodies actually based on the amino acid sequence in the constant region of the heavy chain. Is called as isotypic variation. So type of heavy chain in each immunoglobulin class is immunoglobulin class and the heavy chain type. So now I will tell you about how the classes are differentiated. The IgG is called IgG because they have a gamma heavy chain, whereas IgA has a alpha heavy chain, whereas IgM has a mu heavy chain, and delta D has a delta heavy chain, and IgE has a epsilon heavy chain. So you can just remember the heavy chain by E is epsilon, D is delta, M is mu. The Greek alphabets they are actually.
Yes.
A is alpha and the G is gamma. So variable and the constant, I'll be explaining each about the regions, the variable and the constant region. Each light and heavy chain has a variable and the constant region. What is a variable region actually? A variable region, amino terminal of both light and heavy chain is seen. Therefore, the short form is VL. That is the variable region of the light chain. Light chain I told you is out the outside. You see the one light chain, one variable region and one constant region. But heavy chain will have one variable region and it will have 3 constant regions. So the on the heavy chain it is called variable heavy chain, whereas light chain is called variable light chain. That's a VL and VH. It represents the antigen binding site, and antigen binding site is called as so in the antibody is called paratope. Whereas antigen it is called epitope. Whereas on antibody it is called paratope. Yeah. So next is on this in this variable region, right? On this variable region there is a small area called hypervariable region. Okay, just imagine on the variable region, there's a small area called hypervariable region. It is what is this hypervariable region? Is within the variable region, there are some zones that relatively show higher variability in the amino acid sequence. That's why it's called hypervariable region. They form the antigen binding site. Got it?
Yes, sir.
Yeah. So this you might find where is the paratope? This paratope is on this hypervariable region.
Hypervariable region.
Yeah. So the site on the hypervariable region that makes the actual contact with the epitope of an antigen. The paratope is on the hypervariable region. Hypervariable region is on the variable region, and variable region is on the antibody.
Yes.
So you got it right?
Yes.
Okay. So now there's something called idiotope, antigen determinant of an antibody against which antibodies are generated. Actually, these antibodies are within our body itself, but sometimes there is some where our autoimmunity happens. So there will be autoimmune reaction. That reaction happens because there will be something called idiotope. Idiotope is present on the antibody, which that is antigenic determinant of an antibody against which antibodies are generated. So sometimes there will be on this paratope, there will be an area called idiotope, and this idiotope will causes the stimulation of antibody protection. So this idiotope is already present on an antibody, but still it will stimulate an antibody production. That's why it's called idiotope.
So this is what causes autoimmune diseases or something.
Autoimmune disease also, and it is also example like it's like horse immunoglobulins. No, like if even though immunoglobulin is like sometimes no, the foreign immunoglobulin we used to give for treatment like for like horse pancreas, so it's insulin, right? We used to get insulin
Yes.
from for pigs, or we used to
Pigs
get
and
some
horses.
immunoglobulins from the horse, but they are already
Yes.
antibodies. So when these antibodies are injected into the body, they will have some what para idiotope, and that will cause an immune reaction. Even autoimmunity also this can be a cause. Idiotope can be a cause.
Oh, okay.
Like you said earlier, autoimmunity, this idiotope can be a cause. And even the foreign antibodies injected into our body also
Can
idiotope
also
is a
act
cause.
as an idiotope. Yes.
Okay, so you got it?
Yes, sir.